technical marketing specialist here at GenScript. I received my PhD from the University of Michigan in neuroscience, where my thesis focused on the role of beta-1 integrin, integrin-linked kinase, and caveolin-1 in neuroblastoma cancer. I then worked as a technical support scientist at Ingenuity Systems and then at System Biosciences, where I helped new customers learn to use their products and services available from these two companies. I've now been consulting for GenScript since 2015. Rena Hay and I worked on developing today's webinar for you. Rena is a PhD scientist, senior scientist from R&D at GenScript. Her work is mainly to develop new products to meet all the needs for protein research. She earned her PhD degree from Peking University of China. During her PhD career, her projects mainly focused on the investigation between the host autophagy and pathogen infection. Her studies have made a great contribution for human infectious disease therapies. To further pursue her research career, she started her postdoc research at the Rockefeller University in New York, and she's now been working for GenScript since February 2016. To ensure the sound quality during today's webinar, all of the attendees have been muted. Please feel free to type in your questions in the chat box in the lower right corner throughout the talk, and at the end of the talk we will address all of the questions. So today's webinar is focusing on cancer immunotherapy, advancing and accelerating immuno-oncology research and development. And it consists of 4 parts. First, I'm going to introduce some background information about cancer immunotherapy and further clarify the state and the state of this and the details of 4 different types of immunotherapy. From the future of cancer immunotherapy, we can tell more about the opportunities and challenges in this field. Finally, I'll share some of GenScript's one-stop solution of products for cancer immunotherapy research. We all know that cancer is one of the most serious diseases in the world, and these are just a few statistics for you. First of all, there are over 14 million people diagnosed with cancer every year, and this number is increasing year by year. Half of all men and 1/3 of all women will be diagnosed with some kind of cancer during their lives. And nearly 8.2 million people die of cancer each year. This basically equates to 1 person dying of cancer every 4 seconds. So it's really urgent and important for us to find new and efficient approaches for fighting cancer. We know that our immune system is a protector of our body, basically defending against invasion of bacteria and viruses. That's shown up here in the slide. As it implies, cancer immunotherapy means to treat cancer by using the immune system. The difference here is that the foreign invaders that need to be cleared are actually tumor cells. Typically, oncogenesis leads to the expression of antigens on tumor cells. And these are captured by antigen-presenting cells, also called APCs, which then prime and activate cytotoxic T cells to attack the invading cancer cells. The T cells also release other immune factors like antibodies and cytokines that help to destroy the tumor cells. The traditional cancer therapeutic approaches mainly include surgery, chemotherapy, radiation therapy, and targeted therapies. Surgery is useful at removing tumor masses, but it is possible to leave microscopic amounts of tumor behind that can then go on to form new tumors. Chemotherapy uses generalized medications that inhibit cell division. While useful, it can also target other healthy dividing cells, causing unwanted side effects. Radiation therapy also targets dividing cells, but it can cause unwanted side effects such as burns and also targeting healthy dividing cells. All of these traditional methods are damaging and they have side effects. Number one, they non-specifically and seriously destroy the normal cells. And number two, the cancer is easy to relapse. So it's really urgent to find out new methods that are more efficient and more specific for treating cancer. Cancer immunotherapy really has many benefits. It's powerful in that it attacks the cancer systemically, so that treatment doesn't have to be localized to the primary tumor site. It is specific in that the immune cells can be trained to target the cancer cells instead of other healthy dividing cells. Additionally, as the immune system has the properties of both the innate and humoral immunity, this means that the immune cells are able to learn to respond to the same foreign invader over a long course of time, so the treatment can go on for a long time. And lastly, the cancer immunotherapy is universal in that it can be used for treating any type of cancer. So one question is, why doesn't the body just automatically fight cancer with its own immunity? And one working hypothesis has to do with immunoediting. Immunoediting is characterized by changes in the immunogenicity of tumors due to the anti-tumor response of the immune system. This results in the emergence of immune-resistant variants. And this escape from cancer from immunity has three phases. The first phase shown here on the left is called elimination. During the elimination phase, immunoeffector cells, such as the natural killer cells, with the help of dendritic and CD4 positive T cells, are able to recognize and eliminate tumor cells. As a result of the heterogeneity, however, tumor cells which are less immunogenic are able to escape this immunosurveillance. And these cells are shown here as green cells. This goes on to the second phase, which is called equilibrium. The next step in cancer immunoediting is the equilibrium phase, during which tumor cells that have escaped the elimination phase and have a non-immunogenic phenotype are selected for growth. This is the longest of the three processes in cancer immunoediting and may occur over a period of many years. During this period, new tumor cell variants can emerge with various mutations that further increase the overall resistance to the immune attack. So then during phase 3, called the escape phase, these tumor cell variants, and they're shown as green cells, green ovals here, are selected, and basically they have breached the host organism's immune defenses with various genetic and epigenetic changes, conferring further resistance to immune detection. This diagram shows the whole cycling process of how cancer cells are recognized and interact with T cells, and finally killed by the immune system. It is a complicated and well-regulated process, and there are lots of proteins and genes involved in each of the steps. Random mutations here can cause the escape of tumor cells from early steps to the late steps during the cycling process. For example, during the elimination phase, which includes both the innate and adaptive immune responses to tumor cells, the innate immune response has several effector cells, such as natural killer cells and T cells that are activated by inflammatory cytokines, which are released by the growing tumor cells, and macrophages, and stromal cells also surround the tumor cells. The recruited tumor-infiltrating NK cells and macrophages produce interleukin 12 specifically, and interferon gamma. These kill tumor cells by cytotoxic mechanisms such as perforin, TNF-related apoptosis-inducing ligands, also known as trails, and also with the formation of reactive oxygen species. So as you can see, this whole process has many different points at which regulation could occur. So now I'm going to go through some of the types of cancer immunotherapy that are currently available. So to prevent the escape of cancer cells from the immune system, scientists have focused their efforts on investigating the interaction between cancer cells and the immune system and the escape mechanism. Based on the previous investigations, some approaches have been proven to work within the reactive immune system to target the mutant tumor cells. Most of these have shown a high potential therapeutic market in the future because of great specificity and effectiveness. Here I'd like to show 5 different approaches to fighting cancer by activating the immune system. First, we'll talk about monoclonal antibodies, then cancer vaccines, adoptive T cell transfer, immune checkpoint inhibitors, and then other non-specific immunotherapies. One of the body's natural immune responses to foreign substances is the creation of antibodies specific to antigens found on the surface of invaders and damaged or infected cells. Monoclonal antibodies are man-made antibodies engineered to target specific tumor antigens, and they work in a few different ways. First of all, flagging the targeted cancer cells for destruction. The monoclonal antibody can act as a flag that attaches to parts found only on the surface of specific cancer cells. This marks them for destruction by other immune cells. One example is called CANPath, which is used to treat some patients with chronic lymphocytic leukemia. CANPath binds to the CD52 antigen, which is found on lymphocytes, including leukemia cells. And then once attached, the antibody attracts the immune cells to destroy these cells. A second way is our blocking growth signals and receptors. Some monoclonal antibodies are engineered to block the tools that cancer cells use to grow. These monoclonal antibodies work in a variety of ways, including blocking certain pathways that allow the cancer to thrive, or by destroying the blood supply for the tumor. Cancer cells rely on these blood vessels to deliver oxygen and nutrients, so blocking them prevents the cancer cells from growing. For example, the drug Herceptin is an antibody against the HER2 protein. Breast and stomach cancer cells sometimes have large amounts of this protein on their surface. And when HER is activated, it helps these cells to grow. So Herceptin binds to these proteins and stops them from becoming active. The third type of way that the monoclonal antibodies work are by bispecific antibodies, which are made up of parts of 2 different monoclonal antibodies. So they can attach to 2 different proteins at the same time. One example is called Blincyto, which is used to treat some types of acute lymphocytic leukemia. One part of Blincyto attaches to the CD19 protein, while another part attaches to CD3. This drug then brings the cancer cells and the immune cells together, which is thought to cause the immune system to start attacking the cancer cells. And then lastly, conjugated monoclonal antibodies are monoclonal antibodies that are joined to a chemotherapy drug or to a radioactive particle. In this case, the monoclonal antibody is used as a homing device to take one of these substances directly to the cancer cells. The monoclonal antibody circulates throughout the body until it can find and hook onto the target antigen. It then delivers the toxic substance where it is needed most. And this lessens the damage to normal cells in other parts of the body. Cancer treatment vaccines are different from the vaccines that work against viruses. These vaccines basically try to get the immune system to mount an attack against cancer cells in the body. So instead of preventing disease, they are meant to get the immune system to attack a disease that already exists. Some cancer treatment vaccines are made up of cancer cells, parts of cells, or pure antigens. Sometimes a patient's own immune cells are removed and exposed to these substances in the lab to create the vaccine. Once the vaccine is ready, it's injected into the body to increase the immune response against cancer cells. Cancer vaccines cause the immune system to attack cells with one or more specific antigens. Because the immune system has special cells for memory, it's hoped that the vaccine might continue to work long after it's given. Okay, let's talk about adoptive T cell transfer. Adoptive T cell transfer focuses on changing the body's own T cells to fight cancer. This is a promising new way to get T cells to fight cancer. For this technique, T cells are removed from the patient's blood and genetically altered in the lab to have specific antigen receptors called chimeric antigen receptors, also called CARs, on their surface. These receptors will attach to proteins on the surface of cancer cells. The T cells are then multiplied in the lab and infused back into the patient's blood, where they can now seek out the cancer cells and launch a precise immune attack against them. This type of immunotherapy treatment is still being studied and available only through clinical trials. Studies have shown promise in the treatment of leukemia, lymphoma, metastatic melanoma, neuroblastoma, and synovial cell sarcoma. The most developed CAR T-cell therapy, called CTL-019, was recently designated a breakthrough therapy status for results in clinical trials in the treatment of relapsed and refractory acute lymphoblastic leukemia. This status is intended to help expedite the development of drugs. Immune checkpoint pathways are the connections between the surfaces of immune cells, and specifically between the antigen-presenting cells and T cells, or between T cells and tumor cells. And it's to help regulate the immune response. Some tumor cells have proteins on their surface that attach to activated immune cells that block their function. This connection puts the brakes on the attack and is known as tumor-induced immunosuppression. Understanding how to block certain immune checkpoint pathways is an important focus of current research. Immune checkpoint inhibitors are drugs that block the checkpoint from being engaged, which essentially turns the immune response back on. Immune checkpoint inhibitors are currently being studied for use in the treatment against different cancers. One of the well-known targets is PD-1 and PD-L1. Cancer cells have the ability to make certain molecules appear on the surface, including PD-L1 and PD-L2 of the PD-1 checkpoint pathway. These molecules attach to the PD-1 on the T cells and turn them off, preventing attack of the cancer cell. In addition to researching PD-1 inhibitors, scientists are also researching blocking the PD-L1 molecule. Anti-PD-1, PD-L1 drugs block the connection necessary to engage this protein, allowing the T cells to continue their response against the cancer cells. It has been shown to be highly effective in treating melanoma, non-small cell lung cancer, kidney cancer, head and neck cancers, and Hodgkin's lymphoma. More and more antibody drugs targeting PD-1, PD-L1 are being developed by biopharma companies. CTLA-4 is a protein receptor found on the surface of T cells. When engaged by molecules on the surface of antigen-presenting cells, CTLA-4 is capable of reducing the immune system response. So anti-CTLA-4 antibodies block the connection necessary to engage this protein, allowing the T cells to continue their response against the cancer cells rather than shutting it down. Ipilimumab is one of the antibody drugs targeting CTLA-4. It's proved to extend the overall survival rate of cancer patients. It's also predicted that the combination of PD-1 and CTLA-4 targeting antibodies will highly improve the percentage of survivors from melanoma. These checkpoint pathway blockers stop the negative signals of certain checkpoint pathways, but researchers are also studying ways to increase the positive signals between cells that can cause a stronger immune responses. Clinical trials are also studying the use of both types of treatment at the same time. So let's look into some more about the future of cancer immunotherapy now. Other than PD-1, PD-L1, and CTLA-4, more and more immune checkpoint targets are under investigation, but most immune checkpoint blockade drugs are still early in development, and new targets need more supportive evidence. Although the combination of different immune checkpoint blockers has been predicted to be more potent for cancer therapy, the combination effects are still underdetermined. So to meet the needs of the increasing immune checkpoint research market, a more complete and convenient solution platform is really needed. Non-specific immune stimulation gives the immune system an overall boost by exposing the body to certain molecules that can activate immune cells, such as T cells, causing an increased immune response. This type can be used alone or with other treatments to produce increased and longer-lasting immune responses. Like IL-2, it has numerous effects on the immune system and acts as a general T cell growth factor. It does this by binding to receptors on the surface of T cells, and it has been used in CAR T cell therapy research field. Although cancer treatment by the immune system has shown its advantages, the combination with traditional therapy is predicted to be more attractive and efficient. So in the future, more and more clinical research will focus on combining the combination of all these different approaches. So let's look at what kinds of solutions that GenScript has to offer you. To meet the needs of cancer immunotherapy research, GenScript has developed a series of products for a one-stop platform for cancer immunotherapy, including antibodies, proteins, stable cell lines, and related assays. All we want to do here is to make your research more convenient and easier to perform. As we just talked about immune checkpoint inhibitors, antibodies targeting immune checkpoint targets have proved to have a high activity in preventing tumor cell-caused T cell exhaustion. So immunocheckpoint antibodies are one of the key tools for cancer immunotherapy research. Other commercial antibodies have only been shown to work in binding assays, but GenScript's antibodies have been proved to be highly active in functional assays. This means they can be used both for binding detection assays and also for functional activity assays to study the targeted related signaling pathways. We also provide mouse anti-mouse antibodies, which can be used for preclinical studies to study human antibody drugs in mouse models. The mouse resource-based mouse antibody totally excludes the side effects caused by immunogenicity. GenScript also offers anti-immune checkpoint antibody drugs, which can be used for pharmacokinetic and pharmacodynamic studies. These are useful for preclinical research and antibody drug development studies. By using this kind of antibody, clients could monitor the therapeutic antibodies in samples and detect biotherapeutic antibodies that closely resemble the circulating human immunoglobulin. Here's a list of antibodies that we have already launched. There are different version antibodies targeting several hot immune checkpoint targets like PD-1, PD-L1, CTLA-4, LAG-3, and TIM-3. Most of these antibodies have been used for several different purposes, so that you can choose the most suitable version based upon the application that you're using. So for instance, you can see here that many of these have been used in ELISAs, back studies, and other functional assays. In addition to providing antibodies, GenScript also provides the corresponding proteins, which could be used to modulate immune response functionality, identify binding partners, discover novel drugs, and they can be used for your own antibody production. We also can provide customized proteins if we don't have one off the shelf that is useful for your research. To meet our customers' needs for cell-based in vitro assays, GenScript has developed a series of immune checkpoint stable cell lines. All of these cell lines are validated by FACS in intact cells. The high stability of the target expression ensures that B cells can be passaged up to 15 times, and all the cells are certified to be mycoplasma negative. After creating the immune checkpoint cell lines, each was validated for the cell surface expression marker it expresses. This FACS analysis of human PD-1 expression in HEC293 or CHO cells shows that there is a dose-dependent correlation of expression of PD-1 when the cells are treated with the stimulator drug. Furthermore, a FACS-based binding assay shows a similar result. These cell lines can be used as immunogens for immune checkpoint antibody generation, tools for high-throughput antibody screenings, cell-based immune checkpoint bioassays, in vitro anti-cancer drug screening and profiling, and as tools for establishing in vivo animal models for immune checkpoint studies. GenScript also provides an immune checkpoint assay platform to further accelerate your cancer immunotherapy research. Here I'd like to take CTLA-4 as an example to explain this immune checkpoint assay in more detail. IL-2 is an indicator for T cell and Jurkat cell activation. The more Jurkat cell activation, the more IL-2 is expressed in the system. And Jurkat cells are activated when CD80 expressing CHO cells are co-cultured with the CD28 expressing Jurkat cells. So when CTLA-4 protein was added onto a CHO cell and Jurkat cell co-culture system, the CTLA-4 competed with CD28 to bind to CD80. The less CD80 bound to CD28 in the Jurkat cells, the less the Jurkat cells are activated. When CTLA-4 Fc fusion protein and an anti-CTLA-4 were both added into the CHO CD80 cell and Jurkat cell co-culture system, the anti-CTLA-4 blocked the binding between CTLA-4 fusion and CD80, and the Jurkat cells were activated when CD80 were able to bind CD28. So the right-hand figure here shows the result of the cell-based anti-CTLA-4 activity assay for ipilimumab. The EC50 curves of the two independent screens show that ipilimumab were shown. So that's shown here. And human IgG1 was used as a negative control. So we can see that this system reacts sensitively to the antibody drugs. To summarize the current situation and future challenges of this hot research field, the current research is mainly focusing on uncovering immune pathways manipulated by tumor cells and how modulation of these pathways may restore the body's ability to fight cancer. It's also using immune biomarkers to help identify patients who are more likely to benefit from immuno-oncology therapies, and identifying combination strategies that may activate an effective immune response. Still, there are a lot of challenges for further development. Immuno-oncology is currently led by checkpoint inhibitors. However, as niches become saturated, developers must look to identify novel approaches. Immunotherapy and cross-class combinations are proving to be successful and promising, while it should be based on preclinical science and be aware of the combination toxicity. And lastly, the recent approval of Amgen's oncolytic virus called TVEC for inoperable melanoma is giving hope that there are opportunities beyond T cells. So now I would like to open the questions. And if you have any questions, what you can do is enter them into the question box, which is at the bottom right of your screen. And then as they come in, I will read them and try to answer them. So here's a question. It says, what kind of side effects does cancer immunotherapy have? So cancer immunotherapy sometimes can cause something like an allergic reaction. Possible side effects can include fever, chills, weakness, headache, nausea, vomiting, low blood pressure, and rashes. But compared to other chemotherapeutic drugs, the naked monoclonal antibodies tend to have fewer serious side effects. Thanks for that question. Let's see, here's a question. Is the activity of the recombinant proteins verified or not? Yeah, so the specificity is verified by antibody-based ELISA kits, and then also some of them, like PD-1, PD-L1, and CTLA-4, have also been verified by our assay platform. Okay, here's another question. Are these stable cell lines functionally verified or not? The stable cell lines are mainly used for binding assays and not for functional assays. But we plan on launching a new batch of stable cell lines that have been specifically verified for functional assays. Okay, other than PD-1, PD-L1, and CTLA-4, what other immune checkpoints are higher potential targets for cancer immunotherapies? Okay, so some that come to mind are TIM3, LAG3, and CD40. Any other questions? Okay, I don't see any other questions. So I wanted to thank everybody for coming today. If you do think of a question, please feel free to email us at GenScript. You can find our email address right on our website. Thank you.